record this. Welcome everyone to the metabolism lecture. If you will go to chapter 4 and check out cell respiration, starting on page 132, breaking it all down. We use an analogy in the textbook of an accountant's spreadsheet to figure out where the ATP is going. And I like that analogy. That's why I wrote the book in such a way that we look at it as investment, as payoff, all in terms of how we get energy out of the food nutritions that we consume. Now, I'd like to say that we begin with a nice story. This chapter is on photosynthesis and respiration because they're kind of opposites of each other. Most of you know about photosynthesis. The story in the beginning of chapter 4 starts with an old lady whose dad plants a tree, and it's a touching story about where the energy goes, okay? And it's supposed to be applied to the rest of the chapter. So I would read that story because it's supposed to entice you to read the rest of the chapter. And, you know, it's touching, a little heartbreaking, a little philosophical. So I think you'll find that to be fun. And then we'll move on to so do that on your own when you do read the story. And then let's move on to how energy drives life. I mean, if we think about a cell, what's the difference between a rock and a living creature? You know, it's the life force inside of you. Now, where do you get it? Where do you get that life force? We last left off with nutrition and digestion, right? Where you break down food using enzymes. You took the test on that. And by the way, that part of the test you guys did pretty well on. It's always the respiration that's a bit tougher. But with digestion, you're breaking the food down in your tube and you're making it accessible to be absorbed in your intestines. And so indeed, now we want to know, what do you do with it? What do you do with those digested nutrients in the body? You know, you eat the Twinkie, you eat the ho-ho. Now what happens once it's been absorbed into the body? And we say that it gets metabolized. You know, you hear the word metabolism. You know, fast metabolism sounds good, right? That means you burn down food quick and you don't carry extra weight. And slow metabolism means you eat a Twinkie and then you gain weight, right? So people do have different amounts of metabolism. What is metabolism? The sum total of all reactions in the body, all the reactions. Just add it all up, sum total of all the reactions in the body. And again, if we start with page 132, we move from phase to phase on how we break up that food. There's two parts of metabolism. One is catabolism, another is anabolism. Catabolism, as you can see on the overhead here, is the breakdown of complex molecules to yield energy. So in other words, taking a macromolecule that you've absorbed, let's say, you know, a fat, and breaking it down to yield energy from it. That is catabolism. You know, a car has a catalytic converter. Catalysis, catabolism is all about chopping or breaking down. Car is breaking down gasoline for energy. You are breaking down food for energy. So I give an example. Glycolysis breaks down glucose. If you look on figure 4.13, you can see that the series of reactions in glycolysis are shown, and you're taking glucose in this example. Glucose is what we start off with. So glucose is simple sugar. Glucose is a 6-carbon simple sugar that we break all of our macromolecules, whether you're a protein, whether you're a fat, whether you're a nucleic acid, you're broken into glucose and you are catabolized. That is the way in which we break down food. It's processed until it reaches the glucose level, and then it goes through step 1, glycolysis. Glycolysis is the reaction set that you see on figure 4.13, series of reactions that break that glucose sugar. Again, glucose is a single-ringed monosaccharide sugar, breaks it into 2 3-carbon pyruvic acid or pyruvate molecules, as seen in figure 4.13. All right, so that's catabolism. We're going to talk a lot about catabolism. Something that's often not brought up with metabolism is the anabolism part. That's the buildup of complex molecules to yield energy. The buildup of complex molecules to yield energy. Excuse me, not to yield energy. Sorry about that. The buildup of complex molecules using energy. You have to put in energy to build something up. Just like you got to put in energy to build up a brick house, right? Got to put all the bricks together. You have to put energy in to build up structures in your body. Feel your hair, feel your nails. All of these are constantly synthesized keratin protein, and it requires energy to produce those body structures. All right, that's anabolism. If you hear the word anabolic steroids, that's something people, bodybuilders have taken to build themselves up using these steroid hormones. Right? That's another story for another time. But our example here is converting amino acids to larger proteins. So in other words, making muscle. You want to make muscle, got to have enough protein, eat enough meat, eat enough beans, and the actin and myosin proteins we talked about in ANP1 all have to be built up by anabolism, all right? Some principles you should know about are the exchange of electrons in chemical reactions, known as oxidation-reduction reactions. And what I write here is basically living systems gain and lose electrons spontaneously in reactions that they just carry out. So atoms that gain electrons are known as anions. Atoms that lose electrons are known as cations. Anions become more negative. Cations become more positive because the electron itself is negative, right? So go like this, Leo, grrr. All right, Leo is a lion, grrr is a growl. That's how I remember. Loss of electrons, oxidation, that's Leo. Grr, gain of electrons, reduction, GER. So oxygen is reduced. If a substance is reduced, we say it gains electrons, it gains negativity, One other thing I want to say is losing electrons is the opposite. Lose electrons, lose negativity. In other words, you become more positive. So in general, we say that oxygen oxidizes. It's a taker. It takes electrons. And during reduction, oxygen is reduced. Means it gains electrons. Hydrogens, a golden rule is hydrogens always travel with electrons, H and E. Hydrogen and electrons go together. So whenever you reduce something, you gain electrons and hydrogens together. This is a principle of organic chemistry. All right, just know it as a golden rule. Put it on a flashcard, Leo Gur. And that will help kind of think about how these reactions occur. I'm not going to test you on this slide, but I want you to look at it because it's really the principle behind this chapter. Cell respiration and photosynthesis are opposite. Take a look at photosynthesis. This is the reaction for photosynthesis. Carbon dioxide, you breathe out on a plant. Water, use sunlight energy to make sugar and oxygen. Most of us know this from junior high, maybe even elementary school, right? Photosynthesis is trapping carbon dioxide and making it into sugar using sunlight. The opposite is cell respiration. When we take in food, right, like this, C6H12O6, that's glucose, we use oxygen literally in our mitochondria to produce carbon dioxide that we breathe out, water vapor, and energy. We say that each reaction is opposite of the other. Delta G is the amount of free energy given off. For cell respiration, 686 kilocalories per mole is given off. For photosynthesis, exactly the opposite is absorbed to create the glucose molecule. So these are opposites of each other. Do I want you to memorize delta G? No. But I want you to know that they are opposites, and glucose is oxidized, or in other words, hydrogens and electrons leave the glucose during cell respiration, and instead, carbon dioxide is reduced. That means we reduce it by adding hydrogens to carbon dioxide, and they leave the water in photosynthesis. Again, they are reverse reactions. I just want you to think about that, right? The chloroplast has photosynthesis. and the cytoplasm and the mitochondria have these other reactions called cell respiration. Now, there's three parts of cell respiration. All right, I'm going to show you the three parts. The three parts. First, all right, so what am I talking about? Let's stop there. Cell respiration is like I just said, the taking of a sugar molecule and breaking it down to yield energy. All right. Now the plant has bothered making it. All right, makes it for itself. It's selfish. Makes it for itself, and it itself carries out respiration. All right. The plant itself carries out respiration to break down the very food that it makes. But you eat the plant and you somehow change it, right, through metabolism to sugar, to glucose. And that glucose has to be broken down. How does it get broken down? Cell respiration. Three phases of cell respiration. Number one, glycolysis. Number two, the Krebs cycle. Number three, the electron transport chain. That's what you have to remember. Three parts of breaking down all food in the body. And where does it happen? Glycolysis happens in the cytoplasm, as I write right here. Cytoplasm, that's figure 4.13, happens right in the cytoplasm. All organisms carry out some form of glycolysis, including the bacteria. I don't count viruses because those creatures are not alive. They're parasites, that intracellular parasites. But all what we consider life carries out glycolysis in the Krebs cycle. Glycolysis in the cytoplasm. In the mitochondria, the Krebs cycle and the electron transport chain both occur. All right. Oh, let me tell everyone we are on page 132. All right. Let me just address page 132 for those of you coming, and page 133. And we're going to continue on with the rest of the chapter 4. That's cell respiration. So to reiterate, we've got glycolysis, Krebs, and the electron transport chain. That's what we're going to go over for this hour. And then we're going to talk about how there's different processing for different macromolecules, fats and proteins, for example. So we start with glucose, the splitting of sugar. Again, glucose being sugar, glucose is, and what I want you to do as you go through this, count carbons. I repeat, count carbons. First, we start with what? 6-carbon glucose. And figure 4.13, this is glycolysis. 2 ATP energy molecules are added as what we call the investment phase. Just like a match is added to a fire, you add the 2 ATP and make that match investment to break up the glucose, destabilize it, make it into 2 pyruvates, as you can see on figure 4.13. Now, if you take a look, you find that 4 ATP are actually produced in glycolysis for the 2 ATP that you originally invest. So what is that? A net gain from glycolysis of 2 ATPs. So we're counting carbons and we're counting ATPs. If you notice on figure 4.13, NADH is produced for each of the pyruvates. That's 2 NADHs. NADH is a high energy molecule. Nicotinamide, adenine, dinucleotide. I can't believe I can say that word. But anyway, sounds like a bad word. But that is a high energy molecule. It will be changed into energy later on in the electron transport chain. But 2 are made in glycolysis. Now, if you take a look, I drew the structural formula for pyruvic acid because I just have so much time on my hands. And the pyruvate, also known as pyruvic acid, has to be changed in order to go, see, this is a picture of the mitochondria, in order to go from the cytoplasm to the mitochondria. PA, that says pyruvic acid. In order to get that arrow in and get it into the mitochondria, you've got to do this procedure in your body. The pyruvate loses carbon dioxide, which of course you breathe out. Go like this and say, oh, acetyl-CoA shuttle. You got to say that while you breathe out. Acetyl-CoA shuttle. That's what this is. You're making what we call the acetyl coenzyme A molecule, and you're shuttling it into the mitochondria. And what do we see? NADH is produced, and a carbon dioxide is given off, and the coenzyme A attaches to this part of the pyruvate, making it acetyl coenzyme A. All right? That's an acetyl group. Okay, Now it's able to get into the mitochondria, and in the mitochondria, we carry out something called as the Krebs cycle. There's a couple of names for it, and if you look on figure 4.14, the name I use in the book is the same as I like using on the test, and it's called the Krebs cycle. That's this. Some you call it the TCA cycle, some call it the citric acid cycle, but most commonly known as the Krebs cycle. All right, you're going to go over this when you do micro. You know, Klausner may go into this, may not, if you take micro at Delhi. But ultimately, it's important and taught over and over by different teachers and applied differently in different subjects of biology, because it's the fundamental basis of getting our energy, these three steps: glycolysis, Krebs, electron transport. Okay, so acetyl coenzyme A enters the Krebs cycle. What is the Krebs cycle? A series of enzymes sitting in the mitochondria. Acetyl coenzyme adds to oxaloacetic acid. That may be on the test. Oxaloacetic acid is a 2-carbon, a 4-carbon structure. And acetyl coenzyme A, as you can see, is a 2-carbon structure. And together they form the 6-carbon citrate. And citrate becomes ketoglutarate and blah, blah, blah. Different things happen. And we have lots of ATP, NADH, CO2, and FADH2 produced by the Krebs cycle. Mainly, not even as much ATP is made as NADH. There, for every turn, 3 NADHs are made, and 1 FADH2 is made. What is that? Okay, you're probably asking. Think of when you play cards. When you play cards, if you ever play cards, you play poker. Different hands are worth more. Like 3 of a kind is worth more than 2 pair. 2 pair is more than 1 pair. 3 of a kind is like the NADH. It happens to be worth 3 ATPs. FADH is worth 2 ATPs. ATP is worth 1 ATP. ATP, you know from ANP1, is the energy molecule. Energy is the life force. You could say it's the soul, the spirit, the life force, whatever it is, that's what drives motion and activity and life. So I think what I'm getting at here is this is where the energy is coming out of the food, the Krebs cycle. You have these NADHs and FADH2s that have to be changed into ATP energy. So where does it happen? In the electron transport chain. That is the main function of the electron transport chain, to convert those high energy molecules to ATP. So again, electron transport chain occurs in the cristae, or the inner folds of the mitochondria. It's a collection of molecules embedded in the cristae of the mitochondria. It's in figure 4.15 and 4.16, okay? Those are the molecules. And these are proteins with what we call prosthetic groups that bind to each molecule, and they get reduced and oxidized as the high energy molecule floats down the electron transport chain. All right, and I have a picture of the electron transport chain right here. Let's just skip to right here. NADH enters the chain at a higher level than FADH2. It goes into FMN, FADH2 goes into FES, all right, whatever those are, flavanone, mononucleotide, FES is iron sulfide. That's not really important for you to know. But what you should know is the NADH comes at a higher level than the FADH2, and therefore is worth more ATP energy. Again, NADH is worth 3 ATPs, FADH2 is only worth 2 ATPs. I would call, and I do in the textbook, I call the electron transport chain the bank. It's where you exchange money. You have NADH, it's got to be exchanged into usable form of money, and that's ATP. So various different cytochromes, which are proteins with different heme groups that transfer hydrogens and electrons, will carry the high energy electrons from the NADH and the FADH2 to lower and lower levels. As they go to lower and lower levels, as you see in figure 4.16, you give off energy. All right, an ATP synthetase gives that energy off. And the final electron acceptor, and this is the big reason why you breathe. And you got to ask your parents, why do you breathe? They'll never know this. You know, over the dinner table, your boyfriend, girlfriend, parents, you know, Here's the simple reason. Oxygen is needed as the final electron acceptor of the electron transport chain. If you do not have this electron transport chain, you would die within minutes and that's how cyanide works. Cyanide blocks this electron transport chain, right? Cyanide blocks it and prevents the oxygen from being the final electron acceptor. It chokes up the whole chain. You've got to have this chain to give you life, the energy force, and that's where oxygen comes in. You can think of the mitochondria as many little factories or many little fires that need oxygen or air to obtain the needed energy from each of those mitochondria. All right. When hydrogen adds, or in other words, when hydrogen reduces the oxygen, what happens? H2O, right? We say it's 1/2 of an oxygen. You know, don't get crazy about it. The half. That's just chemically how we represent it. Half of that diatomic oxygen molecule has hydrogen added to it, and water is formed. And that's why when you breathe at a mirror, you will see water vapor come out of your breath, because the oxygen has been changed from gaseous oxygen to water vapor. Here I wrote about cyanide. It blocks the electron's movement from cytochrome A3, which just happens to be a cytochrome right before oxygen, to oxygen. So respiration stops. Fluoride, that I mentioned in another lecture, blocks the electron transport chain as well. It can kill you if you have a ton of it, but mainly it's in small doses in your mouth when you brush to kill the bacteria in this way, blocking the electron transport chain. So let's talk about this picture, the overview of glucose catabolism. This should help you to see how the accountant's spreadsheet brings energy from each part of the process. Okay, let me reiterate. Let's go back, a little review. If you look on the overhead, glucose is broken down how? By glycolysis into 2 pyruvic acids. You net 2 ATPs, so I put the ATPs at the bottom if you look on the figure right there. And you also obtain 2 NADHs from this process, which go to the electron transport chain and yield energy. The 2 pyruvic acids are converted, remember, with the acetyl coenzyme shuttle, and 2 NADs are produced, right? Oh, the 2 acetyl coenzyme A's from 1 glucose go into the Krebs cycle, combine with oxaloacetic acid, and you get, for each one, 2 FADHs for each glucose and 6 NADHs. All right. And now you've got all of these NADHs and FADH2s going into the electron transport chain, and you get 32 ATPs from the ETC, electron transport chain. Krebs gives you a small amount of ATP too. Totally, you got 32, 33, 34, 35, 36. 36 ATP are netted for each glucose molecule. If you're going to get crazy, there are some books that, say it's like between 36 and 38, depending on heat loss. Don't get crazy. Just, 36 ATP are produced per sugar molecule. All right, and that is how we obtain energy. That is why we breathe and have oxygen. I think this is a fascinating lecture. I hope you did as well. Why we breathe and how we derive our life spirit, let's say, our living energy, however you want to call it. But let's talk now about how we, what happens when glucose is very high, when you have a large amount of glucose. We have something called glycogenesis, storage of glucose as glycogen. If you remember, where is that stored? In the liver. When ATP is high, if you eat too many Twinkies, ATP is high, you will store the sugar as glycogen, all right? And then that can later be converted to fats. So everything is not as simple as you eat a certain amount and then you break down a certain amount. Sometimes things back up, sometimes things are in a deficit, and that's what we're here to talk about for the remainder of the lecture. And a little bit about nutrition. If you eat white rice or candy bar, you're tending to eat more simple sugar. When you eat sugar, you know what hormone gets stimulated, right? Insulin. Insulin production causes the sugar to be taken up into the cells and used. So simple sugars, when you eat simple sugars, simple carbohydrates, we call them, monosaccharides, disaccharides, fructose, high fructose corn syrup, you'll have a spike in insulin, and then the sugar will be taken out, and you won't have as steady a pace of energy from that sugar that you ate. You'll have a spike from the candy bar, and then it will go down, all right? As a result, when you exercise and you eat a lot of those simple sugars, you lower your fat use because you're not tapping into your fat reserves. You're just using the high energy product that you ate, like the candy bar. Therefore, we say that you should eat more complex carbohydrates, that is, more polysaccharides, right? Which is like brown rice, wheat bread, not white bread. And of course, all of us love the sweets. The issue is complex carbs take longer to process because you don't just get the sugar, right, like from the candy bar and go right into glycolysis. You have to process it a little longer. So the complex carbs are better for muscle building, maybe more important than protein, to sustain a high-paced exercise, weightlifting, or whatever have you. So for our phys ed majors, it's important to emphasize complex carbs in the diet. Wheat, whole grain, a potato versus just white bread, because there's more processing and more, you follow me, more sustained use of energy, slower use of energy. So like for an hour, you'll use the energy from a, whole grain cereal versus if you eat a candy bar, look, boom, it just jumps up. You have a ton of energy used, and then you get you get tired after like a half an hour. You eat a donut, you're tired after half an hour, but you have a ton of energy at first. You eat something more refined, let's just even talk about a simple egg for breakfast. You're going to use that energy over a longer period than the donut. So the donut feels better because you got a high, right? You ate the donut, you're happy. But then, that's the first 15, 20 minutes, and then you go down the next 45. Whereas you eat the egg, it's not as much of a high, but it sustains you. You can sustain the pace of your exercise more. So that's a little bit about that. Gluconeogenesis occurs when you have too little glucose and you make more sugar from non-carbohydrates. This is why a low-carb diet doesn't really work. You don't have enough carbs, the body will make it in the liver to protect from damage of hypoglycemia from too little sugar. That's called gluconeogenesis. Neo means new, glucose sugar, genesis means to create. So you're making new sugar even though you're on a low-carb diet. There is some evidence that a low-carb diet is good for diabetics because you don't have those spikes, in sugar. You have more sustained use of sugar. So I don't want to totally trash the low-carb diet, the Atkins diet. So that's a way of looking at it. I think what I support is that Neolithic caveman diet where you eat more fruits, vegetables, also known as the Mediterranean diet, and less of the simple sugars and less of the fats and meats, a lower fat diet. And of course, fat has a bad rap. You need a certain amount of fat. We talked about that. Let's look and talk about lipid metabolism right now. How do we break down fat? Fat cannot just go into, glycolysis. It can't do what we said. Neither can proteins. They have to be processed first. So too do the complex carbs that we just talked about, like the whole grain rice. You've got to process it a little. And processing isn't bad, but often in the case of fat, you're getting a lot of energy per gram of fat. Let me give you an example. 1 gram of carbohydrates gives you 4 kilocalories of fat. Of energy. 1 gram of fat gives you 9 kilocalories of energy. So more than double the amount of energy is in a gram of fat than in a gram of protein or in a gram of carb, right? Carb being like pasta, potatoes, etc. All right. How are fats transported in the body? First of all, we talked about, remember, chylomicrons, little fat protein droplets that are moved through the lymph from the villi of the small intestines. and they are brought to the liver, and there is in the liver and then in all cells of the body, lipogenesis and lipolysis. Let's start first with lipolysis, the breaking down of fats. You take a carb, or how does that happen? Okay, when you don't have enough ATP energy, you start breaking down oxaloacetic acid. If you remember, that is the first part of the Krebs cycle. When you don't have enough carbs and you don't have enough fat and you're pretty much starving. In other words, even if you just don't eat for a day, you will start seeing this process happen where the oxaloacetic acid of the Krebs cycle is broken down. And the acetyl coenzyme A that we just talked about cannot get into the Krebs cycle. Therefore, the liver will change the acetyl coenzyme A into ketones. This should say ketones underneath. And ketones are an acid. And that acidity can lower your blood pH. This is what actually kills the anorexic or bulimic, mainly the anorexic. Or anytime someone is starving, it's called metabolic acidosis. You might want to write that word down. I'll write it for you. It's good to know, could be on the boards. Metabolic acidosis equals lower pH due to ketones. Ketones are ketoic acids, all right? So it's interesting to note. So that's what happens. And when we do the urinalysis, this week you had the urinalysis. Wish we could do urinalysis in person. I was going to say maybe at our pizza party, but I don't think that's appropriate. But urinalysis, if you ever stop by my lab when school starts, we can do the urinalysis. I'll give you the urine strip. It's a good way to test what's going on in your body overall, as you know from lab this week. But ketones are in your urine, okay? And ketones indicate that you're starving, that you're somehow breaking down carbs and fats. And this is indeed how we test for it. And of course, if in extreme cases of non-eating, the pH goes down due to the ketones and can kill the person. So it's an interesting point. The opposite happens with lipogenesis, making fat when you eat too much, basically. So in other words, triglyceride synthesis. You're making fats. When there's too much ATP, too much sugar, the acetyl coenzyme A and the glyceraldehyde phosphate can't even get into the Krebs cycle fast enough, right? So the body will make the ATP and glucose into triglycerides. Usually the acetyl coenzyme A gets broken down, right? But instead, with a lot of ATP and the acetyl coenzyme A not even able to get into the Krebs cycle because you're eating too much, it's made into triglycerides. So when you get a blood test, if you have high triglycerides, you're eating too much, basically. So interesting. And look at the mechanism, a general mechanism of breaking down a neutral fat. Now, you know that fats are made of one glycerol combined with 3 fatty acids. You know that from chapter 2 of A&P1, right? So fatty acids, look at all what goes on. Acetyl coenzyme A, NADH, FADH, all these are produced, all these are processed, to make acetyl coenzyme A. All of this processing happens in this line. I don't want to get into the details of what chemical reactions happen, but energy is made. Glycerol is made into glyceraldehyde phosphate. Processing happens to change pyruvic acid, to change the fat into pyruvic acid to get into the Krebs cycle, all right? And then can the Krebs cycle and electron transport happen? So the point behind this slide is that a lot of energy is given off from fat. That you can't just go right into the Krebs cycle or glycolysis. That look at all this. This stuff has to happen, and you're getting more energy from fat. This here is just, and I know it's the handwriting's not neat, but it's exactly what I just said about ketosis. When there's not enough carbs, when you don't have enough food in you, metabolic acidosis happens. And this is metabolic acidosis. I kind of got ahead of myself, a little excited about the topic because it's such an interesting topic. Ketone breath, by the way, smells sweet. In my own clinicals, I remember smelling that in patients. It's like a sweet smell. And it's the sweet smell of starvation, basically. Oxaloacetic acid, the first part of the Krebs cycle, is converted into sugar, which is needed by the body for energy. So you're kind of eating up your own intermediate chemicals of the Krebs cycle. As a result, acetyl coenzyme A cannot get into the Krebs cycle, and the liver converts that acetyl coenzyme A to ketones or organic acids because the oxaloacetic is being used up for food. pH of the blood goes down, increases your breathing rate to force out the CO2, and acidic blood depresses the nervous system and can lead to death. So it can happen with diabetes as well. This is a risk for diabetes because when the cells are not getting enough sugar because there's not enough insulin, this can happen. Diabetic coma. Last but not least, the proteins. We haven't talked a lot about proteins. You know, there's 20 amino acids in our body. Old amino acids have to be replaced and broken down by the liver. We say it's deamination of protein that occurs in the liver. And the excess protein in our diet is also converted to energy or fat. So you can't just have a high protein diet, not gain weight. Basically, it's how much weight gain is basically how much in calorie wise versus how much out due to your activity and metabolism. That is a raw rule that will never change. And you know, portion control is the most important thing for weight balance. Anyway, making along, and you know, Americans have big portions. That's an issue. So I went to McDonald's. They're like, all sodas are the same price. So of course I get a gigantic soda. You know, it's ridiculous. Why not when it's the same price? That's just terrible. But anyway, getting back, how do we break down old amino acids? The answer is in the liver, the NH2 group is removed from the amino group, okay? If you remember that, let me show you the picture here. This is deamination. This is, of course, our last topic. How do we process proteins in the body? That's this last topic. How do we process proteins in the body? Old amino acids you got to get rid of. That's the building block of proteins. We remove the NH2 group from the protein. Now, I just want to tell you, NH2 is dangerous. NH2, look at this, becomes NH3 in the liver. And right in the body, NH3 is ammonia. You don't drink ammonia, do you? No. So that's got to be removed. That's one of the main reasons you urinate, to remove nitrogenous wastes. Where are most of our nitrogens coming from? Proteins. So pyruvic acid or any keto acid intermediate of the Krebs cycle is made into glucose or energy. So you can either make the amino acid portions into energy, you remove the NH2 group and urinate it out, or you can build it back up into something else. So proteins can be either broken down for energy, built up into something else, or the NH2 discarded in the form of urea. NH2 becomes NH3, carbon dioxide combines with it in the liver and forms urea. This picture sums it all up. All right, let's go over this picture yet again. I'm going to put it in blue. This is a picture of the liver. You see, don't laugh. This is a picture. Feel your right side. That's where your liver is. This is a picture of the liver. And right here, this is your amino acid. Now, what are we going to do with the amino acid? I just want to reiterate. You've got to get rid of the NH2. That's going to be on the test. The amino group's got to go. NH2 is a hazard. If your kidney doesn't work or your liver and you can't do this, if you can't remove the nitrogen waste, you die within days. So NH2 becomes NH3. The NH3 combines with carbon dioxide to form urea. Why? Because it's less toxic. NH3 is very toxic. So very quickly, the liver changes the ammonia, or NH3, that is right here, into urea. And here's a little kidney, and it shows urine. It's coming out as urine. Okay, now what else happens to the rest of the amino acid? Either it goes to the Krebs cycle, and you get energy, or it gets recycled, the keto group, that's the rest of the amino acid, into new amino acids and proteins, or something called reverse glycolysis, where you are going to take the amino group and build it up through anabolism into glucose. All right, the opposite of glycolysis. In our lifetime, and I had to look this up because I did not believe it, we eat 500 to 1,000 pounds of protein. I thought we eat way more than that. I mean, when you eat a piece of meat, that's a lot of protein. Apparently, there's not that many grams of protein even in meat and beans and high protein food. So we only eat 500 to 1,000 pounds of protein in a lifetime. That's kind of a weird thought. And you've got to have all of the essential amino acids. There's 8 essential amino acids that you do not make and has to be produced from, has to be taken in from the diet. You've heard of that. The cereal has all essential amino acids. And therefore, like a vegetarian's diet has to have complementary proteins. You've got to have enough proteins in a meal that provide all of the essential amino acids. So for example, you're never going to have corn and rice as a meal. That's not enough. You have to have like cheese on it. Or you have to have beans and rice. The beans have some essential amino acids. The rice has other amino acids. Meat, eggs, cheese have all of the essential amino acids. So those of you, right, that are lactose intolerant, that's the issue. It's you get a lot of your protein from dairy, and if you're lactose intolerant, you got to shy away from that. And obviously, I don't want to put down the vegetarian diet. I think the vegetarian diet is an excellent diet, low fat, high vegetable, high nuts. Nuts have protein, beans have protein. I, of course, cannot do it. I probably could for a while. And I try it the best I can to get away from the meat a bit. Not doing a great job, but sometimes. And it takes creative cooking. You know what I mean by that? You gotta work at it to make healthy meals. And that's a very important, for phys ed majors and for nurses, that you have a heart healthy diet, not just for the heart, but for overall. They say that you have vitamins A, C, and E are your antioxidants, good for cancer, heart disease prevention. Very important. And fruits, grains, nuts. I don't want to be preaching here about diet, especially being a hypocrite that I am. But if you're aware of it, that's half the battle. Then you'll tend to go toward that. So again, complementary proteins in diets lower in dairy and meat are essential. You have to complement and have all essential proteins. Anyway, hopefully you enjoyed the lecture. I'm going to stop the recording and take questions, and we'll go from there. All right.